nice to welcome you on site, Antoine Yver. So basically, been involved in drug development for the past decades across several companies. Could you just give us as a first sentence your background and how you've basically been involved in pharma and drug research and development?
Sure. So number one, thank you for inviting me. And so my background, I trained as a pediatric oncologist here in Paris at a time 50 years ago plus. So at a time where medicine depended less on the ability to analyze and more on the ability to observe and thus match whatever treatment few were available to actual disease, and really based less on knowledge and much more on that understanding what you were treating and why. And then I've been involved in pharma for 35 years, major drugs, major in the sense that they were unique new pharmacology, which if you match them correctly with the actual disease, you actually had a huge impact on patients. And that, these are drugs like Enhertu, Tagrisso, Lapare, but also a few, or Texotere, or Granosite. I mean, I can cite a few. And in all of these drugs, the impact came from the ability to exactly match the pharmacology and its uniqueness to a very well-defined patient's needs, and thus, develop the outcome and the value for patients and thus for society and the companies.
Yeah, and as you mentioned, some of those drugs kind of reshaped the oncology development and the oncology field in general. So could you take one of those examples, like Enhertu or another one, and just give us a bit more detail about what does it mean, matching, understanding the pharmacology, matching to patient biology. So could you illustrate that with one of those examples?
I'll give you two examples. Tagrisso is a drug which was developed, which today, other than COVID drugs, is a drug which was the fastest drug development from the first dose in human to US approval, 2 years, 8 months. Very, very fast. It was designed to be a drug which was so-called for the second line, so the so-called T790M mutation. So it was very specifically designed to that. Highly potent, highly selective, well tolerated, easy to develop. That's why we went very fast. But the true nature of that drug was to actually be extremely selective and extremely potent, which made it actually a very good drug for first-line treatment and not only second-line treatment. And thus, the big potential and the fact that now it is a standard of care for all cancer patients lung cancer patients in the world with VEGFR, which is 25%, is in fact because it is in first line, not only in first line, but in adjuvant, meaning after you remove the cancer, then you give something which never worked in lung cancer and now works in improving. And just because the pharmacology was unique and not what it was designed for. One example. Second example is Enhertu. Enhertu is an ADC, which is a well-known construct. Actually, ADCs were invented, described first in 1913. Nobel Prize winner published. 1958, first dose of an ADC given here in Paris by Georges Maté, combined, rabbit antibodies with methotrexate, ALL, one dose. It worked. And then until 2010, essentially, companies tried to do it. There were some good drugs, but not massively important drugs. And then it really stopped. Japanese took the question, how can we improve? Because it was incomplete drug. They were working, but not incomplete. They took 7 or 8 years to actually refine every single aspect of why complex ADCs were not working. It went from potency, drug, stability, you name it. 7 characteristics. And once you have this, you can translate into a very powerful tool for well-characterized patients. And at a time when nobody saw that, Pfizer was stopping investment in ADC, Roche, Genentech, Seattle, everybody was stopping ADC. This is 2016. Daiichi Sankyo, was sitting on this, didn't know it was important. Because essentially they had deprioritized completely the drug. Even though it was in clinic, there were 5 patients treated when I joined. And then 3 years later, the drug is on the market and completely revived ADC. Why is it successful? Because the pharmacology was correct and we just matched that. If you go back, we press released with my friend Jose Bezalia in 2016. So with 22 patients worth of data, we said that the drug would not be in HER2 high only, but HER2 low, which is really where the big potential of the drug is, which was completely shifting the paradigm of HER2, which is a major marker in breast cancer. And we said that HER2 matters not in terms of being high, but it could be actually almost any breast cancer. As long as there was some degree of HER2, we would be able to treat, and we do. So now the drug is the most effective drug in breast cancer, and it works for every breast cancer, all except the very few, 1 or 2 or 3%, where there is absolutely zero HER2 expression. So again, matching the pharmacology, understanding why it was unique, why it worked, shifting the paradigm if you need to shift the paradigm, and then developing against that. So these are 2 examples on how you do I did it differently or not conventionally, and that essentially created some new standard on how to do things.
So it's basically innovation then coming from the fact to look at the data a bit differently, because data was already there, as you mentioned. Not everything has been reinvented, but it's basically the ability to have this fresh view on the data and know basically what to look for. Is it the way you would phrase that?
Yeah, so it's a bit more. Yeah, yes, the data were there, but it's not only look. So it is to agnostically look. So do not apply a knowledge you have to the data and see, is that matching my knowledge, but to have an agnostic view and try to truly understand what the drug is, what it does uniquely, how the non-clinical model predicts what it does in a non-clinical, how is this portable, meaning relevant to the human setting, how do you translate that into human setting, and what do you observe in human? So it's a whole complex thing. So it's more than just looking differently at the data. It's actually asking different questions in a very agnostic way. What is unique about your drug? What is unique about your pharmacology? What is unique about the patients you're treating, not only the disease, but the patient, and how can you align all three of them, and then you deliver drugs.
Okay, it's super interesting. So basically, there's really this intertwining between the knowledge and the innovation, and how to basically not kind of miss some innovation because being biased basically by the knowledge or by the previous corpus of knowledge.
Yeah, which is something I often say, that knowledge kills innovation because it blinds you to seeing what is different. The other way of saying it is a famous Pasteur quote, which is typically everybody cites Pasteur and Louis Pasteur saying that chance only favors a prepared mind, and that people translate that, I need to be prepared, so I need to know a lot. And whereas the correct quote is, in the field of observation, chance only favors prepared mind. So you have to observe first before you can have chance. It's only by observing in a manner which is agnostic and not about your knowledge. And that really is, in my view, the key to identify the potential and then transform that potential in the field of drug development, obviously, to transform that into an actual drug for patients.
And it made me wonders basically, today, of course, there is lots of new tools bringing knowledge or a partial view of knowledge to people, especially in the pharma industry. Is basically all those new AI tools from, let's say, old school AI models, generative AI models, large language models. So they are bringing yet another layer of knowledge, or at least aggregated knowledge. But how do you see basically the use of those AI models in the pharma field? What, first of all, do you see some especially promising application of those AI models? Do you think they basically fill some specific gap or fall short in terms of clinical application? So what's your view about drug development field in the light of those AI models?
So it's a big question, obviously. I do think that AI, in my simple mind, because I'm anything but that techie, but in my simple mind, I think AI has two huge potential. One, which is tapped currently as we work, which is an efficiency. It enables to be more efficient, to look faster at things that are already there, but you just cannot or do not take the message and then apply. This is true in the clinical development, how to select the patient, whatever you do, from the avatar patients to how to select the site, how to look at the data, how to quality control your data, and so on. All of this. It's useful, it's necessary, it will improve, it will go faster, maybe a little cheaper. I'm not really sure about that because the money is not there. It's in the pocket of people who are actually taking it, which are different. That's one aspect. And when it comes to drug discovery, it helps in merging views and data sets and trying to essentially have, metrics table, in manner which are beyond the typical capacity of a single researcher, a single research team, or a project or company. Definitely, it helps to home onto few things. So but it is an enabler. It's not a disruptor. It's just going faster, better. But when you have a good drug, as I mentioned, you can go super fast without AI. When you understand how your drug works, and you can really, so you don't need AI, but it will definitely improve and will also avoid, better predict toxicity, durability, and so on. So no doubt about it. And the big question is, how can it further improve? I do think that it can further improve mostly by empowering researchers to two things. One is to bring potential meanings in an agnostic way. So exploring more than just a common average knowledge in a manner which is more efficient that one single human being is capable. So exploring different, without hallucination, but exploring different what if it could mean this, and that, number one. Number two, definitely by bringing foundational model about physiology, human physiology, not only the disease and the drug, but the human physiology, how healthy you are. So meaning your state of health, which is different from person to person. And we know that there are big markers of health, CRP, age, but not age, but loneliness, and other big factors which are massively affecting outcomes in disease, but which are completely ignored by everybody just because we cannot influence on them. But they're still extremely important, not by themselves, not as a marker, but of what they are the marker of. What is the human physiology and human state of health, the underlying state of health, the almost thermodynamic balance that you have as a baseline and on which you have a disease. And then move beyond the disease as I have to suppress a disease as this is what good health to I have to control the disease, and good health is also the ability to withstand, injuries or stress, environmental stress or whatever stress, or a disease stress. So this is where AI, by bringing these different views, a potential meaning can actually help identify new targets, and not only at the target at the cell level, or molecular level or genetic level, but actually target of health and not only of disease.
So basically looking at different scales of biology, but also addressing biology from different angles that would be probably beyond humans' mind, being able to both take into account the disease state, but also the physiological state, and putting all of that in the recipe, basically.
Exactly. We humans are able to capture, if somebody walks in a room, capture the mood, the emotion, angriness, being angry, being sad, we capture immediately. But we forget, and we don't take this into account. We also capture if somebody is sick, is tired. Even she would acknowledge, or even be aware. We capture when a woman is pregnant. We capture pregnancy is radiant. We capture all this. So we are able to capture things which have to do with the state of health and not only the state of disease. We're just ignoring it for two reasons. One is because we don't have the granularity, the level of, and the tools, the objective measures. It's only very qualitative. And second is because we also lack the tools to influence it, other than, be healthy, work, and don't drink wine. We don't use the tool, or we don't have the tools yet. But the tools are coming. Pharmacology is coming in that field. But even without the tools, I do think that being able to comprehend this better and at least not ignore, even takes measures as simple as CRP at baseline, or the history of CRP in people. This is the most critical predictor of cancer outcome, of death, is your CRP 10 years ago. Nobody is looking at that. So there's a state of health that AI will be able, the foundational model in physiology and also will help here. And if you can plug that into the data set that you guys are using, trying to help the researcher and the discoverer and the drug developer, that would be terrific.
And then we would move from just being an efficiency and productivity tool, as you mentioned at the beginning, to being a real enabler for more diverse and more innovative discoveries.
Or enricher, you will. I mean, that I truly believe it will enrich our understanding of physiology, of pathophysiology, and thus the ability to change that and thus health. So it will enrich, it will create, it will bring to human understanding measures of health that we can influence and which are today, not within our reach. So it will, and that is very disruptive, as opposed to being an enabler.
And basically, this sounds a lot like a physician answer, and you're basically acting here as a kind of physician researcher, and now taking the pure pharma hat, in a way, in your opinion, to make AI basically a real cornerstone tool within pharma and within pharma industry. What would be for you the main risks or the main potential, basically, on top of being basically this research tool, to make that part of the daily life, basically, of this pharma development? What's missing today, basically?
In other words, embedding AI into the fabrics of pharma, it's obviously an efficiency tool, from the support function to marketing, commercial function, to definitely R&D, in terms of efficiency, no doubt. It has to. Your second point, which is, how can we then further, it should be into, as a critical component of pursuing the white space. In today's age, probably 90+% of targets have been discovered and drugged. So we can improve them, so you can have best in class, but it has been discovered and drugged. And thus, if we can further improve, we have to improve where we are not already doing the same. If we just continue to go after, you'll go into rare disease. True, there are 600 or 700 rare diseases. We can find this and this and that. It's not going to change human health. We'll still suffer from what we suffer and the disease we suffer because we are still incompletely addressing the diseases. It will definitely shift, the scale and scope. And why do I believe this? You're right. I mean, I truly believe that I'm a physician scientist, and in physician scientist, the key word is physician here. And by physician, it means observing scientist. It means asking questions and not being blinded by data, but just like artists are good at one thing, only one thing. They ask you questions. They make every person looking at art ask a question, different question, they ask a question. And physician scientists all about asking the question based on observing and not only on knowing.
Yeah, I love this parallel between our AI tools and platform and a more artistic perspective. And I think it's probably a great way to close that conversation. So thanks a lot for your insights from drug development to AI to help to basically open new avenues to make these AI models really efficient for physicians, for researchers, and within the pharma world. So thanks a lot for those
Thank you. I enjoy. I really do. Thank you.
Thanks a lot.